Hi, everybody. Welcome to New Frontiers in Functional Medicine, where we are interviewing the best minds in functional medicine. And of course, today is no exception. I am very excited to be sitting here with the co-founder of Amazentis. Let me give you his background, and we're going to jump right into some very cool science. Dr. Chris Rinsch is co-founder and president of Amazentis. For more than 20 years, he's been an innovator in the life sciences arena. Before founding Amazentis in 2007, Rinsch worked in venture capital, investing in pioneering life sciences companies in the nutrition space. He also invested in biotech and worked on developing cell-based therapies at the Swiss biotech company Amazentis SA, formerly Modex. Rinsch has authored original publications in top-tier journals such as Nature Medicine, Nature Metabolism, JAMA Open, and many others. And this is focusing on his research on urolithin A, mitochondrial health, muscle function, and joint health. He's also an inventor on several internationally filed and granted patents. He has an MBA from INSEAD in France, a PhD in cell and molecular biology from the University of Lausanne, and a master's in science in biomedical engineering from UT Southwestern Medical Center, and a Bachelor of Science in Engineering from Harvey Mudd College. Welcome to New Frontiers, Chris.
Thanks, Kara. Thanks for having me here today.
You know, your background is really impressive, and it just begs, the question of, a little bit of your origin story. I mean, how you ended up founding Amazentis and really kind of lighting on urolithin A. You've been up to a lot of interesting stuff in your life.
Well, I think the origin of Amazentis comes and timeline comes from my immediate previous experience where I was in venture space looking at nutrition and investing in a lot of different nutrition companies and thinking about health and wellness, sort of looking forward, and how can we have products that are really scientifically based and clinically assessed to actually improve your livelihood, throughout your life, versus a lot of the nutritional products that we have today that essentially it's kind of taking a trust me approach.
Yeah.
And you're really not sure. You take them and you hope for the best. And so that was sort of the origins of, let's take the tools that scientists are using to discover new drugs and apply that to the foods that we eat, and start, taking a deeper dive into what compounds are found in the different foods and sort of linking them to biological pathways, and particularly pathways that are associated with cellular health and well-being and aging as a whole.
That's so interesting. I mean, so when you founded Amazentis, had you already identified urolithin A and just kind of rally your focus around that, or were you looking at many other molecules at that time?
Well, actually, we started looking at it. It was really more of a concept at that time. Let's try and identify those foods and those bioactives in the foods that we eat that could have the biggest impact on health. And so we started exploring. We cast the net and looked at a number of different types of food products, but one of those was the pomegranate. And so that's really how it started. There was a lot of talk around the pomegranate and antioxidant effects of the pomegranate at the time, and so we wanted to dig a little bit deeper to see if this was more than just a marketing story to sell juice, and there was something really behind it. And so we looked at the pomegranate as well as a number of other foods at that time.
It's so impressive. I mean, you guys cast a really wide, well, first of all, you set a really high bar. Let's just make people healthier. In fact, fundamentally, it sounds like you are asking, the question that's on everyone's mind now, which is, how do we slow aging? How do we reverse age? You were really asking these big, lofty questions. And let's apply, I think it's awesome that you take the power of science and you light it onto nutrition. I mean, that's, I think our field, functional medicine, we really embrace that lens. And so, it's exciting for us to see what you've been doing and how you've been thinking.
No, thanks. We were very excited at the onset. And now, I mean, even more excited by what we found and all the work that sort of progressed over since the origins of the company.
Well, I want to jump around. I've got a bunch of questions. There's a lot to talk about here, but I just feel like we might as well go to the origin story, like to discovering Urolithin A, and like let's talk about the lab of Johann Auerwerks. I think I didn't say his last name correctly, but let's just talk about it. Let's just talk about how you guys got Urolithin A.
Well, it really started with this, us taking a look at the pomegranate, and we were really curious to know what was inside of the pomegranate. And so we went through a whole process of actually even purchasing pomegranates, juicing them, understanding what compounds were inside, and then doing this, what we call in science a bio-guided fractionation, which basically means sort of exploding the pomegranate in terms of all the compounds that are found inside of that, and then starting to test those different compounds in different, in the lab on cells to see what type of potential benefits they have. And then we sort of take that to the next step and then take that into mice. And in this case, early on in the company, we were looking at behavioral effects on memory. And this led us down the pathway of looking at a class of compounds that are called the elagitannins that are found in the pomegranate. And more specifically, there was one elagitannin in the pomegranate that's known as punicalagin. It's the most abundant one. And so we started testing that, and we were beginning to see some very interesting effects on actually the behavior and memory in a mouse model of Alzheimer's disease at the time. And so we started from that and started asking ourselves a little bit more questions about, this compound, how is it transformed when you consume it? What is sort of that whole pathway?
Let me ask you that, like, how were you administering it in these animal studies? Were you injecting it? I mean, or were you administering it orally? And why did you start thinking about that? Like, why did you think about the gut systemic interaction or the transformation by the microbiome? What prompted you to think about it with these compounds in pomegranate? Or were you already aware of the transformative microbiome, activating these? So yeah, curious about that.
Well, it's always been about this concept that we wanted to identify different types of bioactives, polyphenols that we could put into food. So we wanted products that were working and that could integrate into your diet, whether it was a food or a supplement. And so in terms of the administration, it has always been through oral administration that we've been delivering, in this case, punicalagin. And to your question about Urolithin A, we had, of course, there was knowledge and there were scientists that had been studying just the metabolism of various foods that you eat, whether they're walnuts, certain types of berries like raspberry and pomegranates in humans, just to see sort of what transpires when you consume these products and the polyphenols and where they end up. And there was this understanding that some people can actually transform, and we'll get that, transform these compounds that are called elagitannins into other compounds that are called urolithins through the gut microflora. And so at that time, no one was really thinking about these metabolites, or we call them postbiotics, as actives, but more as just the body's way of just eliminating the polyphenols that are found in the foods. And so that's sort of that, coming back to your question of how did we land on Urolithin A, was really this, taking this very thoughtful approach as to which compounds might emerge if you were to consume these starting compounds, in this case, puny collagen.
And then, so then you had some, well, you probably were working with mice, and then you were seeing the transformation into the urolithins and other compounds, I'm sure. Yeah. And then
so exactly. So what we started doing is we took that sort of, it was a certain leap of faith at a moment where we said, okay, well, I wonder if these compounds are actually bioactive versus just something that the body is eliminating. And so we then started taking a closer look at them. And so just maybe to go backwards a little bit before we jump into urolithin, what's interesting is that, when you're consuming, whether it's animals or humans, when you're consuming pomegranates or walnuts or raspberries or any of these foods that contain these elagitannins, they're transformed by the gut microflora, so the bacteria that are inside of our gut, into urolithins, which are postbiotic, and in particular urolithin A. But in fact, there's only about, let's say, 30 to 40% of the population that's able to make that transformation. So it's something, and that was what was very interesting when we were studying that and understanding it's not really, you aren't what you eat. You're a combination of, what you eat plus your whole body's ability to transform that and to assimilate those nutrients.
It's kind of extraordinary. So in the animal model, is it similar? Like some mice can make these urolithin A compounds, some mice don't, or to varying degrees of efficiency. And then you had to get in there and look at the compounds individually and see, who's doing the heavy lifting in terms of.
Well, we didn't spend too much time assessing the metabolism of mice, but we quickly, after we started testing some of these, well, in this case, urolithin A on cell models and showing its bioactivity, we quickly focused in on that molecule and to try and understand what it was doing. And so I think one of the first things that we were doing, and this is, you had mentioned the lab of Professor Johan Owerks here at the EPFL. He's a specialist in mitochondrial biology. I provided him a few of these compounds, and he tested that on some of the models that he uses. And he came back and said, what exactly did you give me here? And that was what was really interesting, that he saw this improvement in mitochondria function in these cell models that he hadn't really seen before. It was at a different level. And so one of the areas and one of the ways he studies mitochondria function and aging is using a model that's called the C. elegans model, which is a worm that has a very short lifetime and lifespan. And so by administering bioactive compounds to these worms, you can see their effect on health and longevity by just simply monitoring their lifespan and their prolongation of their lifespan, and also sort of the movement of these worms with time. And so what he saw was that that urolithin A was having this dramatic impact on the lifespan of worms, and also on just the worm's ability to move with time as they got older. And we compare that when you think of these different ways of assessing the lifespan, or different types of interventions that you can make that would improve the lifespan and the health span. And we like to think of things on terms of health span and not lifespan. Yeah. And we'll talk a little bit more about the difference of that. But the caloric restriction, for example, has been shown to improve the lifespan of these worms dramatically by around 50%. But when urolithin A was provided, we were seeing an extension of lifespan by about 45%. And then when you start looking at other compounds that are having an impact on similar pathways, but not identical ones, like resveratrol, for example, you see improvement by about 15%, and that sort of order of magnitude. So this was really something at a different level. And it was that it was these findings in the lab that really caused us to sort of, think about what could this be used for? What type of health benefits should we be looking at? And quickly we started looking at actually skeletal muscle function, because it was so easy to monitor and to measure as animals got older. And animals are, do follow a similar pattern as us humans, in terms of, as they get older, their muscle function declines, they're not as active. And so we started studying that.
Fascinating story. Thank you for unpacking all of it. How extraordinary and how exciting. So basically you had this like caloric restriction mimetic, at least in the C. elegans model. That's amazing, because I think caloric restriction is sort of considered to be really one of the strongest, or one of the strongest interventions to date. And so those early studies must have been just tremendously exciting. And you were extending, obviously, as you pointed out, the worms were moving, and they were high functioning. So health span and lifespan. Right. Just a really neat finding.
Yeah, and I think, going into when we saw it moving into a rodent model, which was so powerful, was to see the translation from different species, going from these C. elegans worms to mice, to see that they were able to, in some of the first studies, we were showing improvement in muscle strength by around 10%, improving in running endurance by over 40%. And just duration that the animals would run. It was very very impressive. And this is what, when we saw this, the translation of these findings and the translation of the biology into mammals, we decided that this is something exciting, that we should actually take it to that next level and investigate that in humans. Yeah,
100%. So I just want to say that, first of all, you guys spent a lot of time in researching. You founded your company in 2007. So I just want to point that out to listeners. You'll see a pretty lengthy bibliography of, careful effort in defining this molecule. The other thing that I wanted to point out, or just ask you, I have a couple questions, so I'll put them out there, is, there are other urolithins, and Urolithin A is the one that you've found to be most potent. And I know sometimes and also, this was the other piece, so I'll throw this in there. We're not all making Urolithin A. I've tested myself through, using your lab, and I make some. I was really excited to see that I make some. However, I don't make a therapeutic amount, because I measured after I actually used your product, after I took the Timeline product, and obviously I was moving a whole lot more out after that. So even when we many of us don't make it, and those of us that do make it, I would say that it's the rare individual that's actually making sufficient quantities to exert the influence that you were seeing. So other Urolithin A compound, other urolithin compounds, and just thinking a little bit about our ability to make a therapeutic amount.
Yeah, sure. So there are a number of different variants of urolithins. As a class, they're called the urolithins, and basically it's just slight modifications of the molecule, and it's due to variations in our gut microbiome. And there are scientists out there that have just focused on trying to understand, what is actually, what percentage of the population can make different variants, and just sort of identifying and describing all those variants. What we the process that we took, that we followed was really to first of all, we focused on urolithin A because it was one of the most commonly seen variants found in those people who are actually able to convert the elagitans or ellagic acid into urolithins. And we've studied basically 4 of the urolithins, this urolithin A, B, C, and D, and what we saw was that urolithin A in a laboratory setting on cells was the most potent, and so and the fact that this was the most predominantly observed in the population, we felt that this was the most appropriate one to look at and take a deeper dive.
Okay, good. Yeah, people have asked me about that, and I know that there is some, biological benefit in the other urolithins. They've been studied, yeah, my understanding was that they are less impactful based on your research.
Yeah.
So from the get-go, you knew that it seemed like urolithin A was working its magic in mitochondria specifically. Is that true, or was that a kind of a journey? And is that why you started to focus on muscle? And then, let's kind of move over into talking about some of the human research.
Yeah, well, it was a journey, I have to say. I mean, when we started, we saw this effect, as I was mentioning, on memory, and then we went to try and understand a little and dig down a little bit deeper to understand how it was working. We saw this effect on improved mitochondrial function, but we had to take that next leap in terms of effort to understand actually what was it doing to improve mitochondrial function. And so this was quite a journey to sort of tease out those mechanisms of action. And this was done in the laboratory of Johan Ulrichs, as we were speaking about before. And I think what was exciting was the discovery that was made there. Together, we basically identified that it was stimulating a process called mitophagy. And mitophagy is part of this process. It's a sort of a subset of a bigger process inside of the cell that's called autophagy. And what this is the cleaning up of cellular waste or garbage and the recycling of it. And so when we think of mitophagy, we think of the cleaning up of damaged mitochondria and allowing those damaged mitochondria to be broken down, recycled, and then healthier mitochondria to be created. So to maintain this high functioning mitochondria level inside of your cells. And so we've been talking here about mitochondria without sort of going into details of what they're doing and how they're helping people. And for those who don't know, yeah, mitochondria are sort of basically the power plant inside of our cells. They're like the little batteries that provide the energy, and that energy is used for all of these cellular processes inside the cell, and keeping the cell functioning. And so when your mitochondria function decline, it's not really good for your cells to start with, but then the tissues that are made up of these cells stop functioning as well as they do normally. But going back to mitophagy and sort of that journey, we performed a lot of studies to try and understand this process, and it was exciting to see that urolithin A stimulates this process of mitophagy, and this basically allows the mitochondria, when they're producing energy, they get damaged, and it allows that damaged mitochondria to be basically sequestered, recycled, and then you have a lot of healthier mitochondria inside. And the results of these studies were published in Nature Medicine, and the reason for that, this was the first compound, a natural one too, and very safe, that was shown to actually induce mitophagy and induce mitophagy in animals when when fed to the animals. So that was a pretty exciting finding.
Really exciting. And, going back to the C. elegans studies, I mean, it explains healthspan and lifespan. I mean, because, healthy mitophagy, the ability to recycle, and it's not just getting rid of the old mitochondria, because then we run out of mitochondria eventually, but it's the renewal journey. I think it's incredibly important to sort of underlay that and highlight it and exclamation point it. This renewal journey is stimulated by the urolithin A molecule, and that's why they lived longer and healthier and moved better, and the mice had better memory, and all of these things, like this extraordinary far-reaching effect. Mitophagy is a hallmark of the breakdown in mitophagy, is a key hallmark of aging. I mean, this drives the aging journey forward. Scientists can go in and mess up mitophagy and basically make whatever they're studying old. So it's just this fundamental process that we need to get right. I mean, it makes sense that you would jump over to looking at muscle because, of course, muscle are dense with mitochondria, and you would really probably, and you said that it was easy to research that organ, but mitochondria are everywhere. Like, it makes sense. The brain is a pretty energy dense organ, and it makes sense that you would see benefit there. And God, I bet some of those early studies when you were exploring, you were seeing some pretty neat stuff mechanistically. Any comments there, or we can just sort of move into?
Yeah, well, I mean, I think that was really what caused us to transition and go on to humans, basically, and start investigating this in humans, and that's been a pretty exciting journey in itself. That's going from a mouse to a human is a big step, and as we know, not everything that shows some type of positive effect in mice will translate into humans.
Probably most fail, right?
Yeah,
but you guys started with something that was consumed, like what was considered a healthy food in humans, and then kind of work backwards.
Yeah, and the nice, I mean, I think the nice thing is we're talking about something, as you said, healthy. This is coming from the pomegranate, and different types of nuts and berries, and the world's population has been consuming this since the dawn of time, and so in terms of the safety profile, you can't really ask for a better safety profile than this, and so that gives you this comfort, at least when you go into humans and you test something in humans. And so we did these first tests, and the first tests were really designed to see the translatability, but also to understand potential dosing levels.
That was my question. Yeah.
So we went through this whole process of testing at different doses, escalating the dose, taking the same approach that even a pharma company would take with a new drug, where you would go in and you would carefully titrate that dose to try and identify the one that's having the right effects and the right benefits. And this is all part of our approach of raising that bar in terms of the level of science to be used on nutritional products. And so that first study, we showed that, and it was very interesting, because it was one month long study, and we were looking at doses, escalating the doses, and we found that 500 milligrams taken daily for 1 month was having an impact on mitochondrial function in the skeletal muscle. So we looked at biopsies. So we took biopsies before the administering the urolithin A to volunteers, and then after a month, and then we looked at the gene expression patterns, and what we saw was that there was an upregulation of mitochondrial genes, and this is clearly linked to an improvement in what they call mitochondrial biogenesis, which is the creation of more mitochondria, and so a hallmark of sort of healthy mitochondria, and we saw other effects in terms of biomarkers that were circulating, And so it was at this point we, in identifying those doses that we said, okay, now we can start looking at, let's say, the physiological impact on muscle at these doses, and giving that over a longer period of time. And so that's when we started to do some studies that were longer in duration, running studies that were 2-4 months long, and looking at the impact of urolithin A after these time periods. And so one of the first studies that we did was in middle-aged, healthy individuals, but who were sedentary and overweight, and we looked after 4 months of administering urolithin A at 500 milligrams, and also 1 gram on a daily basis, and this is where we saw something that was very exciting. We saw that we had an improvement in muscle strength by about 10%. Following this daily administration. And this isn't, mostly when you think of, improving muscle strength, really you need to think about, actually doing some type of physical exercise to do that, and doing that on a repeated basis. But in this case, we're looking at individuals who were sedentary and overweight, and we saw this improvement. And that was really a big breakthrough, and that caused us to also, start to, explore that in other populations and older populations. And we saw after 2 months, in an older population that was 65 and older, looking at people who had a decrease in mitochondrial function at the onset, we saw an improvement in muscle endurance in the hand muscle. And so they call that this first interosseous muscle, and also in the leg muscle after a period of 2 months. And so that was also, a very exciting finding. And, essentially we started to see after a number of different clinical studies, we started seeing, one study after another sort of confirming the similar types of benefits from different angles. So, which was, pretty exciting to see. Yeah.
It seems like you guys just had sort of a ride, just more and more excitement. There must have been some sort of failures or something along the way. We don't have enough time in this podcast, but it just seems like step after step after step was just sort of expanding, how extraordinary this molecule is.
Well, we've been, we've been fortunate to, that actually the science is really solid, and there's, and when you look at in the literature, since we started publishing, there's been a number of other labs out there that have published and have explored the benefits of urolithin A in different animal models, all sort of linked to mitochondrial function. We were talking about it, and you were mentioning it earlier. Mitochondria are found in basically all of our cells, with the exception of the red blood cells. And so, if you improve mitochondrial function, you're going to improve not only your muscle cells, as we were speaking about, we were also talking about brain, your cardiac muscle is another muscle, it's just, and even
lung function,
yeah.
Gastrointestinal function. The cell, the enterocytes turn over like so rapidly. All of these energy demanding.
And so, yeah, it's very exciting. And so right now we've started with muscle, but now we're expanding into new areas. And one of those new areas that we're looking at is immune health. And so this is going to be our first study in immune health, where we're looking at individuals who are taking our product for a month and doing a very in-depth characterization of the impact of urolithin A on immune cells, because there's been some very interesting effects on sort of reversing immune aging in animals. And so we want to explore that in humans and see how that might be beneficial.
Can you speak to any specifics, like what kind of cells you're seeing change?
Yeah, so I mean, I think that for in the human studies, we're in the process of conducting the clinical study, and so we'll have data next year to talk about what's being impacted at the human level. But what we see and this is work that's done with collaborators, we see an effect on T memory cells, an improvement there. And then,
Awesome.
and generally, sort of an impact on the immune cells themselves and this whole immune aging and sort of reversing this immune aging. But this is only I should caveat all of this is all in mice right now, and that's the reason why we're going and we're currently doing this test in a clinical study in humans, and we do all of our clinical studies are double-blind, placebo-controlled, randomized studies with the intention of trying to really understand how these work.
Now, I talked to Anurag, and we were, I mean, you're partnering with a lot of universities. Your research is being conducted on this molecule all over the world in some really interesting applications. I think, well, cardiovascular disease, to your point, I think some neurodegenerative conditions are being investigated, if I'm not mistaken, and these are in humans. Can you speak to some of those partnerships that you're doing?
Sure. So, I mean, I was just mentioning a study that we are doing over in Germany right now on the immune system, and this is something that we're actually doing with the same professors who have published a study last year on mice in the journal Immunity, where they were showing mechanistically how it was improving immune cells. And so we decided to advance that into humans. And you were speaking about brain. Brain, of course, is something very interesting to us. And, anything that we do in humans is all in healthy people. So we're not looking at neurodegeneration. We're looking at the impact of, on healthy people and how it can improve, brain function, cognition, et cetera. And this is something that we plan to do in the future. We're currently not conducting any studies on the brain yet, but it's part of the long list of different types of benefits that you want to improve and prevent from deteriorating as you get older.
Yeah.
So I mean that's what's really exciting there. I think one of the other areas that we've been working on that's very fascinating is going and focusing on administering urolithin A in another way, and that's also topically. And so we've been all about nutrition, and we tend to think about everything that you eat should be improving your health, and that sort of as that way of having an intervention. But topical application is very important for improving your skin health. And
what, can you just give me the backstory on that? I mean, I'm a huge fan. I use the topical Urolithin A, the Timeline product, every day. I'm a huge fan of it. And how did it just make sense to you that a topical application could work? And I mean, what's the backstory on that?
Well, several years ago, we were thinking about, what are all of the different potential applications. And as we were speaking, mitochondria are in all the different cell types. And I thought, it would be interesting if we could put this topically. And so we started sort of exploring that, looking at that on skin cells in vitro to see if we were going to have an impact on skin cells too. And of course, we were starting to see that as well. And so we said, well, let's take a look and see what type of impact we can have when we apply that onto skin. And so we developed some formulations. And you were mentioning that, in one of your earlier podcasts, you had our chief medical officer, Dr. Anurag Singh, and he basically, worked on coming up with different types of clinical studies to evaluate the skin cream and show its different benefits, and taking into account the types of benefits that we were seeing in cells in humans when administered orally, and one of those being an impact on mitochondrial health, and the other that we continue to see is this impact on inflammation, and so we started taking a look at both of those, and so what's fascinating, most people, when they think of skin creams, they think of cosmetics, and we like to think of our product more as skin health, because it's not just about the cosmetic appearance. Of course, that's very important, but it's also about sort of the fundamental skin health, and it all starts at the cellular level. And as we were just speaking earlier about organ and tissue, and that starting at the cellular level, it's the same thing in the skin. And when you can apply it right to your skin, you know right where it's going, and there's no mystery there. So and this way we've been able to study things quite effectively, and look at the effect on inflammation, sort of post UV exposure, and we see an impact on that sort of reduction of inflammation post UV exposure. We've also been able to look at just generally the effect on mitochondrial function and skin cells, and then also the impact that it has on other pathways inside of the cells, including collagen production, et cetera. And then of course the question is, well, do I see anything different when I look in the mirror? And yes, I think that's one of the exciting things too, is that you do see an impact on your skin. You see, I think one of the notable things is you do see an impact on wrinkles in your skin. Not that I would call this a wrinkle cream, but and that's because it's acting at that, fundamentally at the cellular level, and having that impact on the structure and and the health cells. Yeah.
And when do you see this? When do you actually see these changes? Like what kind of turnaround, what kind of time does one need to invest in?
Well, with the products that we've developed, we start to see benefits as early as 2 weeks, which is quite quick when you consider how with respect to the oral nutrition products, that this is changing your cells and getting them to sort of function differently, and the tissues to function differently. It's not something that's overnight. So it does take time and regular application.
That's it's exciting. Yeah, I've been pretty happy with the products myself. I mean, I think that I was aware of your company for quite a while, and just really, wondering, paying attention to some of the science you were doing. And so it's really kind of an honor to, to be connected with you now, and just continuing to participate in learning and sharing the science that you're doing. I have a couple of other just kind of fundamental questions. How did you light on the dose? Actually, so I have a few questions. I'll throw them out there.
Sure.
Why are you limiting to healthy populations? Will you move away from that model? Curious about that. Dosing. So I think a gram tends to be the most efficacious. If I'm not, well, correct me if I'm wrong. And then did you try, were there any side effects at any of the amounts of dosing that you experimented with? Because my experience is then it's incredibly well tolerated. I'm a clinician in practice, and I prescribe this to my patients, and, it's a non-issue for everyone, even some of the most sensitive of individuals that I have.
Yeah, well, maybe I start, with your last question and go backwards. So we were talking about the safety level before. Now, there's just historically, people have been taking different types of foods for, centuries that contain that administer the precursors, these allagitannins that yield urolithin. So and so this is also a very positive thing, in that we see that there's, this is very safe in general, that there's a long history of exposure, and there's a lot of safety. When there's been even publications, not by our company, but by other scientists, academic scientists, where they've looked at women who are consuming pomegranate juice, and they found urolithin A in breast milk. So, I mean, this is something that's been exposed naturally to the population for as long as we can think. And but back to the question of the dosing, we did look at dosing, and we've looked at different dosing levels. 500 milligrams is a dose that we saw after 1 month was having a measurable impact on mitochondrial function. 500 milligrams is also improving muscle function after 4 months. So it's my understanding, and through all the science that we've done, that 500 milligrams is really a dose that you can rely on to see a functional impact, and to experience that functional impact. And so, it's true, we have run a few studies at a higher dose, and sometimes we do run studies at higher doses, and let's say a more in certain populations. For example, we ran a study looking at elite and sub-elite athletes, and we studied that over a period of 1 month, and instead of a period of several months. And so we wanted to understand, what impact we would have on this population. And this is research that has been presented earlier this year. And what we saw was that there was an impact on muscle recovery following these different types of trainings, and in this case, runs that were done over a period of 3 months, and the perceived effort, and then different biomarkers of that were linked to muscle recovery. So I mean, there may be situations where a higher dose may provide or offer benefits if taken in a shorter period of time, but we like to recommend the 500 milligram dose because we've studied that now, and we've seen some very reliable results.
Good. That's great to hear. Yeah. And side effects.
Yeah, we haven't seen any side effects. I mean, we've had now tens of thousands of people who have been taking it as customers, and then we've had hundreds of people who have been taking it as in the context of very carefully studied clinical trials that have been, as I said, double-blind, placebo-controlled studies, where we take a look at all types of panels of biomarkers and in the blood, et cetera, and we have never seen a side effect.
Yeah, it's. I mean, again, this is a molecule we evolved with. It seems like we evolved with that kind of information to sort of direct molecular traffic.
Well, you're right. It's an interesting observation that somehow our intestinal flora has evolved to extract the benefits of these compounds found in the pomegranate and walnuts and other berries, and transform them from these large complex molecules into these simpler structures that have this impact on mitochondria, and that improve mitochondria and that are absorbed into the body. So it's fascinating to see this sort of evolution that we have, and that the microflora has had to be able to to extract those benefits. Yeah, it's
this incredible Gaia hypothesis, the symbiotic interaction of from or where we came evolutionarily. Yeah, it's pretty neat. And it's nice, so we knew everybody, we knew these were superfoods, pomegranates, walnuts, etc. We've known these were superfoods. And as you said, we've been consuming them across cultures for millennia. And so now to apply the scientific lens to it, to actually be able to see at this level what's happening, right down to the microbiome, transforming it and what it's doing systemically, it's just really, it's an exciting place to be in. And it certainly is really validating the importance of nutrition. And
it's this, I mean, it's, it comes back to our earlier parts of our discussion, this, what is that next level of nutrition, this sort of nutrition 2.0, and that's, it's more than just, eating healthy and eating a Mediterranean diet is very important, but, what might be the future of nutrition? And, we all want to maintain ourselves at our peak throughout our life. And, and if we can eat foods with the right type of bioactives that have been studied, and at the right dose to have a specific benefit that sort of map to what our needs are, our health needs are, this is great because it empowers us, and we don't have to wait until we get sick, because most of the conditions of aging are sort of slow degenerative conditions. So I call them degenerative diseases, whether it's sarcopenia or other types of diseases that are degenerative of nature with old age. And so if you can take things earlier that and throughout your life that are preventative in nature and help keep your cells functioning at their peak, there's probably a pretty good chance that that you can slow down that process, and that you can help, live a healthier and more fulfilling life as you get older. And this is back to that whole health span versus lifespan, that you really want to be able to, maintain that your health span at its peak, so you can have a great quality life.
Yeah. Amen to that. Well, Chris, it's been been a great conversation. I mean, this was a lot of fun. I will say, one of my areas, my passion areas has been, epigenetics, and specifically looking at the nutrition influence on the epigenome. And of course, there is Urolithin A has to be an epinutrient rock star, in my opinion. I mean, we know the precursor molecules have influence on the epigenome, and that is, one of the hallmarks of aging, but it's, it's where a lot of my attention has gone. And I look forward to you guys researching that. I would like to, I'll just throw that out there since you're the co-founder, and
we're very interested in this topic too. And so this is something that we do want to take a closer look at in the future, and try and understand, what is the impact in terms of epigenetics if you're taking Urolithin A on a regular basis. And as you said, there probably will be an impact. And so we're interested to understand that.
Well, I look forward to hearing what you guys found. I have no doubt it will be, it'll be exciting and interesting. I mean, we're already seeing research on higher polyphenol diets having broad influence on gene expression via certain epigenetic marks. So it'll be an area I'll keep bugging you on whenever I have an ear from one of you guys.
Fabulous.
Thank you so much for joining me, Chris. It was just a pleasure to meet you and to get to pick your brain today. And I look forward to meeting you in person one of these days.
Sure. Well, thanks so much, Kara, for having me. It's been a fun discussion, and looking forward to seeing you in person one of these days.
To be continued. Okay. Ciao, ciao.
Bye.